So that's GMOs. Now, on to epigenetics. From the video, 30-second review. Epigenetics just means heritable chemical changes to histone proteins. So this gets back to chromatin. DNA is wrapped around the proteins that are histones. Here's a diagram of the 4 different histones that I talked about in this video. H2A, H2B, H3, and H4. 4 different histone proteins. This is all review so far. And those proteins, now what we're looking at are the ends of those protein chains. These are the amino acids. And you notice that it's specific amino acids, which ones, especially, which one amino acid gets most of these chemical modifications? K. What's K? Lysine. So these amino acids in the proteins that are the histones that DNA wraps around to organize itself in our nuclei have lots of lysines. And those lysines can become chemically modified by the addition of acetyl groups and methyl groups. And depending on whether or not your histone proteins that your DNA is wrapped around are acetylated or methylated, different things happen to gene expression. That's the big picture idea. And the table below is something I stole from Wikipedia. You can't see the citation. Oh, there you go. Now you can see the citation. Wikipedia is okay to use. It's totally fine. I use it too. So the big picture is that acetylation, down here in the bottom, is usually corresponding to active gene expression. So if your histones are acetylated, your genes are those genes in the region are turned on, so to speak, active, being transcribed, translations happening, and so forth. And by opposite effect, methylation, especially trimethylation, is usually, not always, but usually correlated with repression. So histone acetylation, genes are on. Histones methylated, genes are turned off. And that happens how? What's happening when you acetylate or methylate histones? How is that affecting gene expression? What's the direct link, the mechanism? It's a little bit more than how the DNA interacts with the histones, but it's how the histones interact with each other. So it's chromatin structure.
They condense together?
Pardon?
They condense together?
They condense. So when things are repressed, when chromatin is condensed, tightly bound, all the histone proteins interacting with each other tightly, gene expression is usually turned off. So in that sense, we could also add in here that trimethylation usually results in heterochromatin formation. That's the tightly condensed DNA proteins. And acetylation usually produces euchromatin, or the open, accessible DNA protein structure, where it's easy for proteins to come in and transcribe genes. And don't be surprised, I will not be surprised, if somebody wins the Nobel Prize for discovering this. It was about a decade and a half ago that the histone code, which is what this is called, you see up there in quotes, was initially discovered that these modifications to histone proteins could actually change gene expression. Huge breakthrough in our understanding of how genes are regulated, especially because these are not mutations. Acetylation and methylation doesn't change whether or not you've got A's, C's, T's, or G's at specific spots in specific genes, but these chemical changes, nevertheless, affect us by changing gene expression, by changing transcription, therefore by changing the amount of proteins that are produced. So you get changes in phenotypes, not by changes in DNA sequence. So that kind of breaks the rule. This is why it was so cool. It kind of breaks the rule of genotype to phenotype. Your DNA sequence produces your trait. because now it's more than just the sequence of DNA we have to worry about, it's whether or not the histones that are bound to that gene, or that gene is bound to, are acetylated or methylated. So that will also control whether or not the gene can be transcribed. So that's the, if you like the prettier picture, there's your pretty picture.